are going to be small molecules that are responsible for either having an excitatory or an inhibitory effect at our postsynaptic cell. And so these are going to vary between synapse and might have different functions within different areas of the body. So the same neurotransmitter will always be released at a particular synapse and then will be quickly removed from the synaptic cleft. So let's go through some of the most common neurotransmitters that we have. So acetylcholine, this is going to activate muscles and is a major neurotransmitter for the autonomic nervous system. So this can be both positive, excitatory, and inhibitory. Dopamine is a neurotransmitter that we find in the central nervous system and is important for reward motivated behaviors. And then in the peripheral nervous system, it inhibits norepinephrine in blood vessels and is a vasodilator. And also will decrease gastrointestinal mobility. So this one too is both excitatory and inhibitory. Epinephrine and norepinephrine, these are going to be for fight or flight responses, and so both of these are going to be positive, excitatory. Serotonin, this is a neurotransmitter that's involved in feeling happy, and so for mood, appetite, and sleep. Histamine, this one is found mostly in the hypothalamus, and it increases wakefulness and inhibits sleep. Glycine, this is predominantly an inhibitory neurotransmitter in the central nervous system, and it helps to improve sleep quality. Glutamate, this is primarily an excitatory neurotransmitter, and it's important for learning and memory. Aspartate is very similar to glutamate, and so it also is excitatory. And then GABA, gamma aminobutyric acid, this one is primarily an inhibitory neurotransmitter that we find within the brain. So these classic neurotransmitters in this table, so a big difference between neurotransmitters and neuropeptides are the fact that neurotransmitters tend to be smaller and they act very quickly. And so again, some can be excitatory and some can be inhibitory. Neuropeptides, on the other hand, tend to be larger molecules and they tend to be more slow acting. And so you see some familiar names, I'm sure, like insulin is considered to be a neuropeptide. Beta endorphins, so these are responsible for getting that runner's high, and also are given with epidurals. And then also substance P, so that has to do with pain. And then oxytocin is important for, it's called the love hormone or the cuddling hormone. And then also some of these, like CCK, somatostatin, and gastrin, we find within the GI tract, digestive tract. When we look at differences between neurotransmitters and neuropeptides, again, one of them is size. So neurotransmitters tend to be smaller in size and only have one amino acid or one small chemical, whereas the neuropeptides tend to be 2 to 20 amino acids in length. So neurotransmitters, because they're so small, can be synthesized right at the synaptic knob, whereas neuropeptides tend to be synthesized in the cell body at the endoplasmic reticulum in the Golgi and then have to be transported. So they're stored in synaptic vesicles for the classic neurotransmitters, and then in large vesicles in the axon terminal that for neuropeptides. So both are going to be released though at the axon terminal. And again, the neurotransmitters tend to have a rapid response, whereas neuropeptides tend to be slower responses. And then site of action, both are going to be in the postsynaptic cell. Okay, and their effect then is for the neurotransmitters is to alter potential of the postsynaptic cell by opening ion channels, whereas with neuropeptides, these can either increase or decrease the effectiveness of the long-term changes in the neurotransmitter or the postsynaptic cell. So drugs are ways in which can affect the neurotransmitter and neuropeptide. And so one example of alteration is by the synthesis of, say, neurotransmitter, how much of transport neurotransmitter occurs, or with neuropeptides, how much of the neurotransmitter that's stored or released can affect, or drugs can affect that. Also modifying their interactions with the postsynaptic receptors, so you can block them or you can enhance them by keeping channels open for longer durations. And so that can lead to changes in tolerance as well. Also, the neurotransmitter reuptake or destruction of them can be influenced by drugs. So if neurotransmitters are broken down, that would lead to like withdrawal symptoms, or then reuptake. If you inhibit that or increase that, then you can also affect chemistry of the brain as well. And then also having a decrease in the amount of neurotransmitter and then replacing it with some type of substitute may not have the exact same conformational changes, and so that could lead to long-term effects as well. So examples of drugs. So with cocaine, this blocks the reuptake of neurotransmitter of dopamine at the presynaptic terminal. So meaning that dopamine is going to be bound to the receptors on the postsynaptic cell for longer, thus allowing more ions to come in. And so over time, though, individuals who do cocaine need more and more because they need to keep those channels open for longer to get that same type of feeling or response. Strychnine, this competes with inhibitory neurotransmitter of glycine at the postsynaptic cell, and so it affects then the motor nerves of the spinal cord, and then which control muscle contractions. And so what happens then too is that there's an increase in excitatory neurotransmitters that are bound to the receptors, and so this will cause an excitatory response within our muscles. So we'll have like spasms of muscle contractions, and then eventually this will cause death though by asphyxiation. And then tetanus toxin, this prevents the release of inhibitory neurotransmitter GABA, and this also affects the skeletal muscle, kind of similar in fashion, where our cells, our muscle cells are overly excited and then lead to motor neurons constantly firing and then eventually leading to death.